Hello and welcome to this webinar titled Current Progress in Approaches for the Safety Assessment of Extractables and Leachables, hosted by Biopharma Asia magazine and presented by Dr. Kim Lee of Amgen and Dr. Erica Tulo of West Pharmaceutical Services. My name is Usp and I will be the moderator. Now, before we begin, I would just like to remind our viewers that there will be a live question and answer session at the end of the presentation. Audience members can send in their questions at any time during the presentation via the Questions tab located at the bottom of your screen, and we will go through these at the end of the webinar. Now, please allow me to introduce you to our first speaker. Dr. Kim Lee is a board-certified toxicologist with broad experience in R&D and operations. She joined Amgen in 2004 and is currently the lead toxicologist on the Extractables and Leachables programs on container closure systems, drug delivery devices, and single-use bioprocess systems. Kim is the chair of the Extractables and Leachables Safety Information Exchange Consortium and, as a member of the Association for the Advancement of Medical Instrumentation Biological Evaluation Committee, Dr. Kim Lee serves as the UX expert to ICO TC 194. Dr. Lee will be doing a presentation titled Current Progress in Approaches for the Safety Assessment of Extractables and Leachables. Welcome, Dr. Kim Lee.
Thank you, Usp, for the kind introduction. Today, I would like to talk to you about the current progress in the assessment of extractables and leachables in pharmaceutical products and medical devices. I will share the challenges we face with combination products, more specifically, a combination of a biologic delivered by an injectable device. I will discuss the relationship between Kramer classification and adverse outcome pathway, which is emerging as a powerful tool for toxicology assessment. I will show how Pqri and in silico tool can be used to quickly screen chemical compounds of concern. I will discuss the concept of action thresholds by applying Kramer classification and adverse outcome pathway. I will wrap up with the points to consider. In 2016, FDA issued guidance on the use of ISO 10993-1, commonly known as the Guidance on Biocompatibility Evaluation. The exact title is shown here on the slide. The guidance was years in the making, addressing extensive comments from pharmaceutical and device industries. It was clear to the agency that there was a desire to move away from the prescriptive checkbox approach to focus more on risk management, to streamline biocompatibility testing, and to address chemicals of concern. The most exciting aspect of the guidance is that it provides a framework for risk management process and encourages the use of chemical characterization of device materials and toxicology assessment of leachable substances as justification to reduce testing requirements. The guidance lists the toxicity studies required depending on the nature and the intended use of the device. The testing includes in vitro and in vivo studies covering significant toxicity endpoints listed on this slide. Of these, the toxicity endpoints highlighted in blue are likely available from open literature and toxicology databases. However, it is the agency's expectation that in vitro and in vivo testing will still be necessary for the remaining endpoints. These include cytotoxicity, local irritation, sensitization, implantation, and hemobiocompatibility. The new guidance asks that we do not simply check boxes for routine tests. Rather, it asks that we understand the clinically relevant attributes in the development of a device. Now, this includes device design, the components, the materials of construction, and the manufacturing processes. We also need to understand how the device is used in the clinical setting, the anatomical location, of the frequency and the contact duration. In this risk management framework, a robust justification can be made for no new testing if the device and components have a long history of safe use, the device and components are chemically and physically well characterized, and absence of new biocompatibility concerns. The new guidance encourages the use of chemical characterization to enable toxicology assessments. In vitro and in vivo biocompatibility testing, polar and nonpolar vehicles and solvents are used to prepare extracts for testing. Examples of vehicles include saline, cottonseed oil, etc. And these vehicles interfere with the chemical analyses of leachable substances. In addition, the levels of leachable substances in the extracts are often far too low to reach toxicity endpoints. Alternatively, device materials can be chemically characterized to improve toxicology assessment of leachable substances. These substances can be identified and quantified, thus allowing for exposure estimates. Literature review can also be leveraged for data-rich compounds, and in silico tools can be used to predict significant toxicity endpoints. Despite the risk management framework, there are still significant challenges for drug-device combination products, most specifically for a biologic delivered in an injectable device. First, protein therapeutics are susceptible to interactions with reactive chemical compounds. The interaction may lead to structural modifications with potential impact to product quality and safety. Second, delivery devices are complex in design, configuration, and materials of construction. Third, manufacturing and processing of materials often requires sterilization, which may result in degradation products. Sterilization by gamma irradiation, steam, or ethylene oxide leads to the formation of degradation products. These may be fragments of polymeric materials and reactive decomposition products of intentional additives used in the production of polymers. These degradation products are reported as tentatively identified structures because there are simply no reference materials to verify the identity or quantitation. To complicate this further, they may be detectable only under certain conditions, depending on the extraction solvents and analytical techniques. As decomposition products, these tentatively identified structures are often reactive, unstudied chemical compounds that pose significant challenges to toxicology assessments. Why should we even care about tentatively identified structures? First, the detection of a TIS during screening materials will inform on the risk mitigation, such as alternative sourcing of materials. Second, the detection of a TIS in a drug product on stability will require considerations for specification limits at recommended storage conditions until expiry. Therefore, a tentatively identified structure is not to be taken lightly, because there is often limited or no toxicity information on a TIS compound, and silico predictions play a critical role. If the risk is considered acceptable for device development, the compound may be monitored as a potential leachable in a final protein formulation with a final device design. For data-rich compounds, the safety assessment is fairly straightforward. However, TIS compounds are often data poor and therefore pose significant challenges, both safety and quality assessments. Safety assessments are further ahead than quality assessments. Indeed, safety assessments are maturing toward best practices. This slide shows the key publications on the safety assessment of extractables and leachables in many product categories and dosage forms, namely the orally inhaled nasal drug products, the parenteral and ophthalmic drug products, biotechnology products, as well as pharmaceuticals and biologics. Quality assessments have a long journey ahead. Protein therapeutics, in particular, are at risk of structural modification by interaction with reactive decomposition products. Such interactions may impact product quality, safety, and efficacy. The formation of protein adducts and aggregates poses significant challenges to formulation chemists, risk assessment toxicologists, immunologists, and clinicians. Safety evaluations typically examine the inherent toxicity of the identified leachable substances, the exposure assessment, and the risk based on toxicity and exposure. On the other hand, quality evaluations examine the impact to the protein itself, namely the protein purity, potency, TKPD, efficacy, critical quality attributes, and stability upon expiry. So, is there a link between safety and quality assessments? The answer is yes. The answer lies in the toxicologist toolbox, and the hidden treasure is in the threshold of toxicological concern of TTC values. In general, toxicology assessments consider two dimensions of risk, namely cancer risk and non-cancer risk. For cancer risk, ICH M7 provides guidance on DNA-reactive mutagenic impurities. For non-cancer risk, the Kramer classification is excellent in predicting systemic toxicity. The classification helps to rank compounds into low, intermediate, and high toxicity. The Kramer Class 3 compounds are most revealing because of the presence of the reactive functional groups. The TTC for Kramer Class 3 compounds is 90 micrograms a day. Please make a note of this because this lays the foundation for the adverse outcome pathway and the action threshold, which I will now transition to. Kramer Class 3 structures are known for their reactive functional groups. Examples of these classical reactive functional groups include cyanyl, N-nitrosyl, quaternary N-epoxides, etc. So what is the significance of these reactive functional groups? The significance lies in the propensity to interact with critical cellular targets, especially DNA and proteins. The adverse outcome pathway is emerging as a powerful tool for toxicology assessments. The pathway describes a series of key events that lead up to an adverse outcome. The series of key events begins at the molecular level when a chemical forms covalent binding with the target. I've listed the AOP references here in the footnote, and the most recent one is a 2018 OECD User's Handbook supplement to the original guidance document. The schematic here illustrates the simplicity of this science and risk-based approach. If we can identify the reactive compound, we can mitigate the risk of covalent binding with protein and the subsequent steps of adduct formation and a significant toxicity, including immunogenicity. Software programs have been developed based on structure-activity relationship of thousands of data-rich compounds. Free for download and open access are ToxTree and QSAR Toolbox. These programs are most popular for predicting mutagenic activity and useful for implementation with ICH guidance on the DNA-reactive immunogenic impurities. However, these programs are underutilized for predicting protein binding alerts. Of these two programs, I find Toxtree to be more user-friendly. Each module is rule-based, supported by the references cited here on this slide. You recall that in general toxicology assessments, there are two dimensions, cancer risk and non-cancer risk. For biologics, we need to consider a third dimension, the risk of protein modification. We propose to use Toxtree as a pragmatic tool to capture all three dimensions. Within each dimension, we leverage literature toxicology information as much as possible, a threshold concept, and a read-across techniques to address data gaps. For the dimension with protein modification, it is important to understand other interaction mechanisms. The compounds that pose highest risk of covalent binding of proteins are the microacceptors, shift-based formers, acylating agents, compounds with nucleophilic substitution, and transition metals. In Toxtree, the module on protein binding quickly classifies the compound structures with these alerts. In our experience with extraction studies, we have encountered these reactive compounds. The examples are shown here on this slide. The concept of adverse outcome pathway has been applied to skin sensitization, cholestasis, liver fibrosis, and liver steatosis. Skin sensitization was among the first pathways described in detail. The adverse outcome pathway for a toxicity endpoint requires in vitro and in vivo data that describe the series of events at the molecular, cellular, organ, and organism levels. The pathway is described here for skin sensitization. The molecular interaction is the initiating event when a reactive chemical covalently binds to skin proteins, and that is heptanization. The subsequent events involve activation of dendritic cells with expression of inflammatory cytokines, mobilization of activated dendritic cells to lymph nodes, and the clinical manifestation of allergic contact dermatitis. This schematic illustrates the series of events that lead up to allergic contact dermatitis. The blue boxes on top describe the successive involvement of skin proteins, dendritic cells, keratinocytes, lymph nodes, leading up to clinical manifestation. In the second row on the far left, the oval in yellow illustrates the initiating molecular event. And on the far right, the oval in red represents the adverse outcome. Now that I have explained Kramer classification, adverse outcome pathway, and the application to skin sensitization, how does it all help with safety and quality assessment for biologic delivery device combination products? Well, for biologics, the product quality may be impacted by lower levels of leachable substances relative to those that may impact patient safety. Simply put, product quality has a lower threshold than patient safety. Therefore, a threshold that protects product quality will also protect patient safety. The concept of action threshold was first published in 2015, designed as a trigger for quality investigations. The premise for the derivation of action threshold is the Kramer class 3 TTC of 90 micrograms a day. Therefore, if we apply a modifying factor of 10 to reduce the risk of interaction, and that is covalent binding, then the action threshold can be estimated at 10 micrograms a day or per dose, assuming daily exposure for a lifetime. The citation for the publication on action threshold concept and its application is shown in a footnote here. As we wrap up this webinar, I'd like to share with you the points to consider. In the absence of harmonized guidance on E&L impurities, consider all safety assessment approaches with science and risk-based justification. Consider screening of E&L impurities with reactive functional groups that may form covalent bonds with proteins. Focus on the chemical compounds of concern with clinical relevance, including systemic and local toxicity. Consider chemical characterization and toxicology assessment in lieu of certain in vitro and in vivo biocompatibility tests. I'd like to acknowledge my mentors and also colleagues who have given me valuable insights from their areas of expertise. These include my colleagues at Amgen, my mentors, including FDA CBER, Ingrid Markovic, Dr. Tony Meisler, who is now at AstraZeneca, Dr. Dennis Jenke, who is now at Triad Scientific Solutions, and Diane Pesky, who is with West Pharmaceutical Services. For those who like to read up on the citations, I have provided full references here on the next few pages. With that, I thank you for your attention, and now I'd like to pass the floor back to Afzal.
Thank you, Dr. Lee. Now again, I would like to remind our viewers that there will be a live question and answer session at the end of the presentation, and that audience members can send in your questions at any time during the presentation via the questions tab located at the bottom of your screen, and we will go through these at the end of the presentation. Now, please allow me to introduce you to our second speaker. Dr. Erica Tulo has over 9 years of experience in extractable and leachable analysis of container closure and delivery systems and devices. She currently serves as Technology Manager for Extractables and Leachables at West Pharmaceutical Services, where the focus of her role is to collaborate across industry to further scientific knowledge and determine the most current testing technology in extractables and leachables. She is an active participant in the International Pharmaceutical Aerosol Consortium on Regulation and Science Working Group. She is also a member of the Healthcare Businesswomen's Association and is a 2018 mentee with the Forum of Executive Women. Dr. Tullo will be doing a presentation titled A Practical Approach to Extractables and Leachables. Welcome, Dr. Tullo.
Thank you, Afzal, for the nice introduction. Hello, everyone. In today's presentation, we will discuss 4 extractable and leachable related topics. The first topic that we are going to discuss are the Usp Chapters 1663 and 1664. We will then touch on some key points from the Pqri PODP working group regarding extractable and leachable studies. For the third topic, we will walk through the different stages in designing E&L studies as part of a successful program. And lastly, we will discuss a case study. However, before we jump into the considerations around E&L testing, I wanted to start with this overview slide, which focuses on fit for purpose in qualifying and using a primary packaging system. The reason I want to point this out is to note that E&L is not the only factor that needs to be considered in choosing the appropriate system. In proving suitability for use, you must consider protection, compatibility, safety, and performance as part of the qualification process. Additionally, there is a need to understand how critical aspects are controlled as part of the ongoing routine supply. Now, E&L is part of what is needed as part of the safety aspect. Kim did a great job of delving into the toxicology aspect of extractables and leachables, and by the end of this webinar, you will have a much more comprehensive understanding of how to put together a scientifically sound approach to E&L testing. Per USP 1663, extractable compounds are organic and inorganic chemical entities that are released from a pharmaceutical packaging or delivery system and into an extraction solvent under laboratory conditions. These compounds, or substances derived from these compounds, are considered to be potential leachables since they have the potential to leach into the drug product under normal conditions and become leachable compounds. Leachables, on the other hand, are either organic or inorganic chemical entities that are present in a packaged drug product because they have migrated into the packaged drug product from a packaging or delivery system, packaging component, or packaging material of construction under normal conditions of storage and use, or during drug product stability studies. As we will see in the upcoming slides, E&L is not a checkbox exercise, but rather more of a journey. While there are general guidances that govern this type of testing, none of the guidances offer step-by-step instructions on how to perform this type of work. It is important to perform a thorough risk assessment and then to design appropriate studies, since regulatory agencies demand proof of the safety, quality, and efficacy of the drug product and packaging material. This, in turn, leads to patient safety, which is our utmost concern. Let's take a closer look at USP Chapters 1663 and 1664. First, I'd like to point out that the USP states that chapters above 1,000 are for information and contain no standards, tests, assays, or other mandatory specifications. Both 1663 and 1664, being over 1,000, fall into this category. They are both informational general chapters. 1663 presents a framework for the design, justification, and execution of an extractables assessment for pharmaceutical packaging and delivery systems, while 1664 focuses on the framework for the design, justification, and implementation of a drug product leachables assessment. Per 1663, extractables assessments can be used for various reasons, a few of which I've listed on this slide. Extractables assessments can be used to characterize packaging, delivery systems, and components as in a material characterization. They can be used to establish the worst-case potential leachables profile, which helps to facilitate leachable studies. Appropriate extractable assessments allow for better understanding of the effects of manufacturing processes on packaging components and their potential leachables. This is very important as it is known that different processes such as sterilization can alter the extractables profile. Assessments can also facilitate the establishment of qualitative and quantitative leachables to extractables correlations. This is helpful in being able to determine which component a leachable may be coming from. Lastly, extractables assessments allow for assessment of patient exposure to chemical entities. The USP 1663 takes into consideration several factors for when generating the extract. These factors include the chemical nature of the extracting medium, the extraction time and temperature, the extraction technique, and the extraction stoichiometry. And when I speak of stoichiometry, I'm just referring to the surface area of the test article relative to the volume of the extracting medium, as well as the actual physical state of the material when it is extracted. Once the extracts are generated, the chapter then breaks down the processes involved in characterizing the extract into four main areas: scouting, discovery, identification, and quantitation. Scouting involves using analytical techniques such as pH or NVR that provide information regarding bulk chemical properties of organic and inorganic entities. The discovery stage involves testing the extract for and finding individual organic or inorganic chemical entities. Possible techniques at this stage include GCFID or MS, as well as LCUV or MS. Identification of an extractable can be accomplished by either structural analysis or qualitative analysis. Instrumentation in this category includes mass spectrometry. Quantitation is typically based upon the instrument response on an individual compound relative to an authentic reference standard. Note that all the techniques that I mentioned are not all-inclusive, and if you'd like that all-inclusive list, please go to the USP 1663 for additional techniques. Also, it is important to note that these techniques can overlap. For instance, mass spec can be used for both the discovery, identification, and quantitation stages. Moving on to 1664, this chapter states that leachable studies can be used within a leachables assessment to facilitate the timely development of safe and effective drug products, to identify trends in drug product shelf life studies of leachable accumulation, to facilitate change control processes for drug packaging delivery systems, and to facilitate investigations into the origin of identified leachables whose presence causes out-of-specification results for the marketed drug. In addition to USP Chapters 1663 and 1664, the Parenteral and Ophthalmic Working Group of the Product Quality Research Institute, or Pqri, has also discussed best practices for extractables and leachables. This is our second topic, and I would like to take the next couple of minutes to go over the table, as it really provides a good overview of the different studies. At the material characterization stage is really when you start screening of material candidates or the components. These compounds that are found during this stage are classified as potential leachables. You want to establish the material composition as well as general biocompatibility at this stage. You also want to employ rigorous, broad-based extraction and testing methods so that you can really learn as much about the components as possible. At this stage, you also get semi-quantitative assessment for toxicological alerts. And the outcome of this stage is either that materials are approved for further consideration or rejected and new materials are evaluated. Moving on to the simulation study, this study establishes extractables as worst-case leachables, and these compounds that come from this study are considered to be probable leachables. You use accelerated conditions designed to mimic worst-case exposure, and this is where you look at the overall system overall, and you have to really justify your use of simulating solvents and extraction conditions. The safety risk assessment of all extractables above the Analytical Evaluation Threshold, or AET, is calculated. Also, this stage can be used to delineate target leachables for the further method development and validation work. At the outcome of this stage, extractables that are deemed to have an acceptable safety risk are not considered further. Target leachables are to be assessed then via the Leachable study. And then at the Leachable stage, that is really when you have confirmed leachables. You establish the actual accumulation of target leachables in the actual drug product. And the drug product is tested under actual conditions of use and shelf life, and you have to perform your safety risk assessment of all targeted leachables. The outcome of this stage is that the safety risk for all leachables has been ascertained, and the packaging deemed to be acceptable, or not. Now, for the third topic that we are going to discuss today, we've put together a roadmap to help guide the process of getting from extractables to leachables. The first stage includes pre-planning and component selection. The second stage is assessment and evaluation of that data. The third stage includes taking the data from the previous stage and performing a risk assessment. And the last stage is Leachables Evaluation. Let's go more in depth in each of these stages. In the first step, pre-planning and component selection. What information am I able to obtain from my vendor as to material characteristics and safety? Some vendors may offer extractable information, and also some vendors may have certifications of additives of concern. It is important to look at this information and see if it is complete information. Does the material that you're looking at have inherent properties to be hazardous? This falls back also to the extractable information that you're able to gather, as well as any regulatory certificates of additives of concern. You also want to take a look at the formulation characteristics. Hopefully the vendor can aid in this information, and you want to take this information fully evaluated, and from this point you can hopefully select components to be used in your container closure system or delivery device. The second stage is assessment and evaluation, and this is where you assess the extractable information and design fit-for-purpose studies. Once you've gathered all of the information, you have to evaluate it. What information is available to help evaluate patient risk? And what is missing? So have all compounds been evaluated? For example, it's very important at this stage of the game to make sure that you have a full understanding of the components used. So you want to make sure that the compounds that have been analyzed have been looked for volatile compounds, semi-volatile compounds, inorganics, as well as non-volatile organic compounds. Has the testing been completed in a solvent similar to the drug product? And what is the drug product contact, time and temperature, and how can that increase or decrease patient risk? Was the information that you have available to you sufficient to prove that the extraction was an efficient extraction? And then also, what is missing based on what the vendor provided? Also, you want to take into consideration, was the component or system tested in its final process state? As I mentioned earlier, any processing step, such as sterilization, can alter the extractables profile. So once you have all the information available, you really need to make the decision. What additional evaluations, if any, are needed? You also want to consider all systems that come in contact with the product during its life cycle. This includes any raw material containers, processing equipment, intermediate containers, packaging equipment, as well as the final container closure system and the delivery device. If a leachable compound is detected in the drug product, it is important to have an understanding of the extractables profiles of these various containers and equipment so that the compound can more easily be traced back to where it came from. Some vendors may provide a potential extractable list for their materials. It's important to note that these lists are theoretical in nature, and they are based on information about the raw materials and possibly the manufacturing process. While they can be helpful in the determination of feasible targets or for the identification of unknown compounds detected during the extraction, it is very important to note that they should not be used in place of extractables data, because potential of migration, as well as some of the processes, may not be used as a factor of what goes on the list. An initial qualitative, semi-quantitative determination of the types of chemical species that can be extracted from a material under extreme conditions that are designed to be more rigorous than anything the material would experience under use. So this is when you want to focus on the individual components of the system. And it's important to note that while you do want a rigorous extraction, you don't want something that will cause the material to degrade. Use of multiple solvents of various polarities and solvent properties are typically recommended for this study. You can also use of known and surrogate standards to obtain semi-quantitative data. And from this information, you can also get identification and estimation of target compounds. In designing of these studies, you want to take into consideration several different factors, such as solvents. If you have a low or high pH drug product, you may want to consider a high or low pH water. There's also isopropanol or an isopropanol water 50-50 mix. Hexane, as well as other different solvents, can be considered. You also want to take a look at the preparation. This is very dependent on what the container closure system or material of interest is. There's soaking, sonication, reflux, microwave, as well as sealed vessel extractions. For example, if your drug product is only in contact with your container closure system for 24 hours at room temperature, a reflux extraction may be too aggressive for that type of container. Techniques that you want to consider when doing these types of studies. It's important to get a good overall picture of the types of compounds that can come out of these materials. So you may want to look at LCMS for non-volatile organic compounds, or GCMS direct injection for semi-volatile organic compounds, or GCM Headspace for volatile organic compounds. You also want to take a look at inorganic compounds. And the techniques that are typically associated with inorganic compounds are ICP, either OES or MS for extractable elements, or ion chromatography for ionic species. Another type of study during this stage is a simulation study. So the simulation study is really designed with the drug product of interest and packaging system in mind. It simulates realistic worst-case conditions to have the same propensity to extract as the drug formulation, drug placebo, or other stimulants. If you simulate properly and design the study correctly, it really is a good indication of probable leachables that you'll see down the road in your drug product. You also want to make sure that you do spike and recovery types of experiments on extractables of interest prior to the simulation to be sure that you can detect and quantitate at a low enough level for meaningful results. You also want to establish extractables as worst-case leachables. As part of the simulation study, you can use accelerated conditions designed to mimic worst-case exposure, and you have to really take into consideration the simulating solvents and extraction conditions that you are using. You also want to make sure at this stage that you're performing a safety risk assessment of all extractables above the analytical evaluation threshold. And you can really use these results of any potential or probable leachables that you find to then target leachables in your drug product matrix and perform method development and validation. The outcome of a simulation study is that extractables that are deemed to have an acceptable safety risk are considered further, and target leachables are to be assessed via leachable study. The third stage is the risk assessment. So once you've gathered all of that information, you really want to take a close look at that information, evaluate it, and organize all the data based on the risk ranking. Now, I know several companies may perform their own type of risk assessment, but it is important to evaluate the extractables highlighting analytes of concern, such as any special case compounds present, the solubility or potential toxicity of the compounds. Your assessment is completed for the specific drug product of interest and container closure system. So you want to take into account the dosing regime as well as the product shelf life. When talking about the analytical evaluation threshold, that is a calculation which gives you a number at the level at or above which an extractable should be identified for potential tox assessment. The SCT is a variable that is used in that calculation. The Pqri recommends an SCT for orally inhaled and nasal drug products of 0.15 micrograms per day. They've also recommended an SCT of 1.5 for parenteral products. And when do you calculate this analytical evaluation threshold? Well, in order to do it, you need appropriate information on the system, such as dosing and volume of the drug product. So therefore, once you have that information, it can be calculated during the extractable stage, the simulation stage, or during the leachable study. And the final stage is the Leachables Evaluation. This is where you're going to develop and validate Leachables methods and monitor them over the shelf life of the drug product. In looking at this chromatogram, it is evident why it is important to fully understand the component or system extractables using clean solvents before testing the drug product. Some excipients, such as twine, can cause chromatographic interferences. It would be very easy to miss an extractable peak, potential leachable, or leachable compound in this chromatogram if the appropriate studies were not done prior to analyzing the drug product. When talking about a Leachables evaluation, it is really specific to the drug product. You want a sensitive as well as targeted method development. So you want to take the compounds that you found in either the extractable study or the simulation studies that you've deemed to be of importance and develop and validate a method for those specific compounds in the specific drug matrix. You need robust methods to discover also the unexpected compounds of leachable compounds that may surface throughout the shelf life of the study. And this is also when you perform method optimization and validation. It is also important to note that Leachables evaluation needs to take place over the shelf life of the study. Some customers decide to do accelerated testing, but that is not always indicative of the full shelf life of the product. So it's important to, if you are doing accelerated testing, to also do studies over the shelf life of the product. And it is also important to note that a system found acceptable for one drug product is not automatically assumed to be appropriate for another. So methods should be validated at the Leachables evaluation stage. An unvalidated method is not valuable in the eyes of a regulatory agency. Also, the detection level should be meaningful. If you choose too high of a detection level, the data will not be pertinent to patient safety. However, if you choose too low of a level, analytical detection may be challenging. It's also important to take into account multiple lots of drug products should be evaluated. This really accounts for a variability. And we have seen cases where you do see variability from lot to lot, so it is important to take that into consideration. So what happens if you do detect a leachable in your drug product? Well, let's remember that there is no inert packaging. At this stage, you want to consult with a toxicologist if a toxicologist has not already been consulted. And anything can be toxic in high enough doses. They will assess the chemical, quantity found, and dosing regime of the drug, and go from there. Now, for the fourth and last topic of the day, let's take a closer look at this case study. A bromobutyl elastomer was reflux extracted in water, isopropanol, and hexane for a period of 4 hours, and then the resultant extracts were analyzed by a variety of techniques. This slide shows the total ion chromatograms from the direct injection GCMS analysis. As expected, there were both more and higher amounts of compounds detected in the hexane extract. The peak at approximately, also just to note, the peak at approximately 27 minutes is the internal standard. And this slide presents the direct injection GCMS total ion chromatograms of the reflux extracts from a butyl elastomer, where we see the same behavior, that there are both more and higher amounts of compounds detected in the hexane extract. One of the other techniques in the analysis of the butyl elastomers was Headspace Gcms. While this is somewhat difficult to see, the blue top chromatogram is the sample and the bottom black line is the blank. So there were some volatile organic compounds detected in this sample. In this case, to narrow down the list of extractable compounds that were detected, a simulation study was performed on the full container closure system with the butyl stoppers. The matrix was a buffered sucrose solution that simulated the drug product, and the vial stopper systems were stored for approximately 20 months, inverted at 5 degrees C. As you can see, no compounds were detected in the sample that were not also detected in the control solution. The Headspace analysis was a little bit of a different story, though. That same solution was also analyzed by Headspace by heating it to 80 degrees for 30 minutes. And as you can see here, 4 peaks were detected in the sample solution. Fortunately, in this case, these 4 peaks were easily traced back to the extractable study. So based upon these results, a leachable method was developed for each of the 4 compounds detected by Headspace GCMS during the simulation study. Only one of the 4 compounds was detected above the quantitation limit in the analysis of the aged drug product using the validated method. A toxicity assessment was then performed to evaluate the impact of the levels to the drug product. The levels were found to be below the PDE, or permitted daily exposure, and were deemed acceptable. In conclusion, it is important to fully assess each stage of the roadmap. At the pre-planning and component selection stage, it's really important to evaluate the available vendor information and literature searches to select appropriate materials. This includes material characterization reports, theoretical material extractables, or any formulation characteristics that the vendor can provide. The second stage is where you want to assess and evaluate and design any additional fit-for-purpose studies. This would include extractable studies specific to the components or specific to the drug product, including migration studies and simulation studies. The third stage, you want to evaluate and organize all the data based on the risk ranking. It's at this stage you focus on what compounds do you want to focus on for leachable method development and validation. And in the final stage, that's where you want to develop and validate leachables methods for specific compounds in the specific drug matrix. It is important to design appropriate studies based upon the guiding principles that we discussed today so that meaningful results can be achieved. This is critical not only for the safety, quality, and efficacy of the drug product and packaging material, but also for patient safety. And with that, I would like to turn it back to Estelle.
Thank you, Dr. Tulo. We will now begin the question and answer segment of the webinar. And once again, I would like to remind the audience that you can still send in your questions via the questions tab located at the bottom of your screen. Now, our first question is for Dr. Kim Lee. What are examples of leachable substances with skin irritation or skin sensitization potential?
Thank you for the question. For biologics, we have not really seen too many skin irritants or sensitization, but what we have seen are quartz, and these are quaternary ammonium compounds, surfactants from cleaning of the equipment. Hopefully that helps.
Great, thank you, Kim. Now, our next question is for Dr. Tulo. is there ever a situation in which you would perform a simulation study over a material characterization?
That's a good question, and I really think it boils down to your risk assessment. If you think you have appropriate information or enough information on the components of the system to be able to target compounds for the simulation type study, then yes, you could probably move right into the simulation study. However, as I've shown, it really is important that you have information on your components so that you can appropriately pick those targets. Also, depending on your simulation matrix, it may interfere chromatographically and you may miss compounds. So that's why it really is important to design a materials characterization before a simulation study. However, if the justification exists that you have enough extractables information, then yes, you can move right into the simulation study.
Thank you, Erica. Now, our next question is for Dr. Kim Lee. So the Pqri has proposed a safety concern threshold of 5 micrograms a day for skin sensitizers for parenteral and ophthalmic drug products. And the question is, how does the Pqri approach differ from use of adverse outcome pathway?
Thank you so much for the question. The Pqri originally based the 5 micrograms a day for skin sensitizers on respiratory irritants because that was really the purpose of the thresholds for orally inhaled nasal drug products. However, as Pqri continues with recommendations from OINDP to parenteral and ophthalmic drug products, they've included other compounds, including, for example, the food allergens, as well as the fragrance materials. And looking at all those allergens, they come down to a very conservative number, and it's still 5 micrograms a day. In contrast, the AOP is based on mechanism, the fact that Kramer classification and also AOP have already been built around thousands and thousands of chemical compounds. We did not delve into those individual compounds or different classes, and yet it's mechanistic because it's based on the very first molecular initiating event. So fortunately, the Pqri threshold of 5 and the AOP of 10 are pretty similar. In all likelihood, they are both quite conservative.
Thank you for that, Dr. Lee. Now, our next question is for Dr. Tulo. What solvents does USP 1663 state to use for extractable studies?
Since 1663 is a general chapter, it doesn't come right out and say exactly what solvents to use. It does list solvents as possibilities, such as water, low pH water, high pH. There is a table in that chapter that lists some possible extracting media depending on what your packaging component is, but it doesn't tell you exactly which ones to use. So that too really is based upon your risk assessment, the container closure system, and the shelf life of the product. It does also mention in 1663 that if the goal of the extractables assessment is a materials characterization, then simulating a drug product vehicle is both unnecessary and undesirable, since these goals require qualitative and quantitatively efficient extractions. So while it does offer guidance in 1663, it does not tell you exactly what needs to be done.
Great. Thank you, Dr. Tulo. Now, our next question is as follows. Is it allowed to use the 1.5 micrograms per day limit for, and in brackets they put unknown?
I can take a look. I can try that question. First of all, the 1.5 micrograms per day is the ICH-M7 TTC for unknowns. there is a caveat in there, and that is you must exclude the possibility of the cohort of concern for using that. The cohort of concern are those compounds such as aflatoxin, nitrosamines, and those have been very well studied. They're not really unknown. So those compounds also have much lower thresholds than the 1.5 micrograms per day. So that is a caveat there if you use 1.5 microgram per day as the limit for unknown.
Thank you for that, Dr. Lee. Now, our next question is a question about accelerated study versus shelf life leachable studies. And the question is, have you ever, I believe they're trying to say, have you ever had any cases where leachables were reported in shelf life leachable studies which was not seen in an accelerated study?
Yes, I can start on that one. The short answer is yes, I have. That's why it's important to do leachables testing over the entire shelf life of the product, because it is very drug product system related, and the interaction between the drug product and system. It is important to study them at the condition, the actual condition that they are being stored. So I have seen cases where you have a leachable over the course of the actual shelf life of the study, but you do not see it in the accelerated study.
Thank you, Dr. Tulo. Now, our next question is, during the early phase of drug development, when dosing regimen is not undefined yet, how do you determine your AET value?
I can jump in on this one as well. You really can't at this stage of the game. The analytical evaluation threshold, you do need your dosing regimen, how many doses per container that you're going to use. So, so per the equation, you can't calculate it at this stage of the game if you don't know your dosing regimen.
Okay, thank you. Now, our next question is, what do you consider acceptable spiked recovery range? And in brackets, they have 50 to 150%, 80 to 120%, etc. Does all spiked compounds meet the same recovery criterion?
I can take this one as well. And this is one that we could probably sit here and talk about and have a very good discussion around. Typical ranges would be 80 to 120%. I have seen some mass spectrometry methods where the recovery range is 70 to 130%. So if you want to go with the typical accepted approach, or the typical accepted recovery is 80 to 120. But I think there's always exceptions that lie outside of that range. And if you design the study where you're happy that if you detected at 70%, you know that you are at least seeing 70% of the compound, you can kind of calculate from there. I mean, it's very important that it's sometimes more so important that you are able to detect the compound versus what level you're detecting, and then you can kind of back calculate, if you will, what a worst case amount of the compound would be. But yeah, so the generally acceptable is 80 to 120, but there are always cases where you may not be able to meet that, just be based on the analytical technique or the matrix, the compound in the matrix that you're using.
Thank you, Dr. Tulo. Now, our next question is as follows. How do you evaluate controlled extractable study data with reflux for 4 hours in water? For example, to bracket your aqueous DP, say WFI diluent with 25 degrees storage temperature up to 3 years.
Yeah. So I'm not quite sure I fully understand the question. But if you're looking at a 4-hour reflux, that really is to get a material characterization on the component. If you wanted to perform a simulation type of study, that's when you could do the Arrhenius equation for accelerated packaging and storage conditions. And that's when you could really predict about how long and what temperature, how high you could go temperature-wise, that you would need to perform a simulation study to bracket that 25-degree storage temperature up to 3 years.
Great. Thank you, Dr. Tulo. Now, our next question, it's a bit of a long one, so it goes as follows. Pqri expected to test multiple lots of materials and multiple lots of DP for EL correlation. It specifically asked to test lots of materials that were not built into drug products. The question is, if there is a leachable compound that tends to completely leaching out, there is a risk to establish the quantitative correlation with leachables from the drug products. There is a possibility that levels of this compound could vary from lot to lot, and the level of this compound may be lower in the unused lots.
I like to say that yes, a lot-to-lot variability for trace analysis, especially leachable studies, is not uncommon. But I would look to Dr. Tulo to give more explicit answers.
No, no, Kim, I do agree that you can see lot-to-lot variability. I apologize. I'm rereading the question because I'm not sure I completely understand what is being asked.
Okay, so Dr. Tullo, what we will do is we can answer that question via email later on after the webinar, and we can move on to the next question, which is, if your product is hydrophobic oil, is it necessary to use other than hexane for extractables?
I've seen clients make the justification that it is not necessary to use other solvents other than hexane, simply because they are not strong enough solvents. So you gather the data, but in the end you know that they're not strong enough to mimic the hydrophobic oil. So is it really meaningful data? So I have seen clients argue that it is not necessary to use other solvents. However, looking at the full picture, if you ever intend to use that product on a different type of drug product, or I should say if you ever plan to use that component on a different type of drug product, you may want to evaluate other solvents. I would say it also depends on maybe the percentage of the oil, whether or not you'd want to use other solvents. So there's a couple different things that you'd want to think about when making that decision.
Great, okay. And we have a question here that might actually be a repeat question, but I'll read it out anyway. Is the actual drug product used in accelerated study and shelf life study? And have you seen leachable detected in shelf life study that was not found in accelerated study?
Yes, I believe we did touch on that one, and the answer is yes, that I have seen compounds come out in the shelf life study versus the accelerated study.
Okay, so our next question, okay, this is a bit of a long one. So for SU systems, guidelines indicate that filtration should be as close as possible to filling. Flush bags are employed to remove the leachables and extractables at the beginning, but not during the process as they are generated. Thus, if double filtration is carried out into an XL SU tank, the concentration will be diluted down to acceptable levels, and there will be no need for flush bags which contain product, and they put waste in brackets. Thus, the question is now, can this approach be taken in the risk evaluation of extractables?
Just give me, if you could please, just give me a few minutes, or a few seconds really, to reread the question.
Sure, go ahead.
The answer to that question is yes. Really what the regulatory agencies are asking for is a thorough risk assessment and why you chose to do certain things. So if you can prove that the concentration of an extractable would go down to an acceptable level due to the process, then I do feel that is an acceptable approach to be taken in the risk evaluation of an extractable compound.
Great. Thank you, Dr. Tulo. Now, our next question is, have you had a successful case where your simulation study shows no leachables and gained regulatory approval without performing leachable studies?
Unfortunately, my answer to this one is really not going to be a good answer. I've seen both cases. I have seen a case where a customer did not have any compounds come out in the simulation study, and they got a comment back from the regulatory agency still expecting to see leachable information. And that was tricky because then it was kind of like, okay, well, what do you target for in your leachable work? I've also seen it the other way, though. So I've seen it both ways. So unfortunately, that's not a very good answer.
But that's very true in real life, depending on how strong your justification is to the regulatory agency.
Okay, thank you, Dr. Lee and Dr. Tulo, for your input on that question. We have time for a couple more questions. The next one is, can you address the challenge of detecting at the AET for high dosage products and large volume area test articles?
That is a very difficult question to answer, and may be one that we'd want to do over email as well.
Sure. Okay. And just as a quick reminder while we're on that topic, if there are any questions that don't get answered today because we run out of time, we will email answers to any questions that we did not have time to answer or that Erica or Kim would like a bit more time to answer. So with that being said, our final question for today, it seems like, is as follows. Is a worst-case extractable study necessary if USP 661.1 is fulfilled?
Let's see. Could just give me 1 minute. Yes, so 661 addresses plastic packaging systems and their materials of construction. So this too may be one left for email, but the short answer would be this only addresses the plastic packaging systems. So if you're using anything else that's not plastic, you still want to evaluate a full study.
Okay. Thank you again, Dr. Tulo. Now, that is unfortunately all the time we have left. Thank you for your answers, Dr. Tulo and Dr. Lee. And just as another quick reminder, we will email answers to any questions that weren't answered today on the webinar, or if we didn't have time to answer them, we will answer them via email. I would like to take this opportunity to thank our presenters, Dr. Kim Lee and Dr. Erica Tulo, for sharing their knowledge with us. And audiences can view this webinar on demand by visiting biopharma-asia.com. Thank you, everyone.